Good morning, everybody, and welcome to another Friday, but this is a very special Friday for two reasons. This is our last seminar for the term, and also we reserve the last day for our amazing students. So the last but not the least, in fact, the most important in my view, because we're talking about the future of the ocean and the world. So thank you all for agreeing to present today. Is going to be really tight. With students, there will be 5 presentations in total, 2 from our master students and 3 from PhD students. And the timing is 5 minutes per master student and 10 minutes per PhD student. So this is going to be quite fast, and I hope you're all ready to take off with them. I have a long kind of bio here. I'm going to be really quick because of time. So our first speaker will be Samantha Ramirez. Sorry for the messing up your name. And she completed her undergraduate degree at the University of Gulf. She's working with Dr. Jordan Rosenfeld, and she's going to talk to us about her work, which is modeling the impact of human development on water quality and hypoxia. So that's the first talk. This will be followed by our second speakers, and there are 2. They will share their 5 minutes, so this is a bit challenging, but they will do it. Kate Mussett and Kasey Stirling are the 2. They will share that time, and they're supervised by our new faculty, who is no more new, Dr. Andrea, right? So her master students, and they're going to talk about a framework they are developing for the co-production and for co-producing a biocultural framework for fish habitats. So that will be exciting to hear. Samantha.
Okay, hello everyone. Thank you for attending this talk today. So my name is Samantha Ramirez, and I will be talking to you today about my proposed research for my master's program. I will be modeling the impact of human development on water quality and hypoxia. Today, I will talk to you first about land use and climate change impacts on hypoxia and water temperature, how this impacts fish, modeling stream metabolism, and estimating current and future impacts to stream fish. So human development, more specifically land use, global warming, and flow regulation, have many consequential impacts on stream ecosystems, some being increased nutrient loading and light availability, and reduced flows. There are many different indicators of water quality, but for the purposes of this study, water quality will be defined as water temperature and dissolved oxygen concentration, where extremely low concentration is known as hypoxia. So what are the impacts of hypoxia and high water temperatures on fish? We know that it can reduce fish immunity to disease, influence fish growth, induce migration, and may also affect reproductive success. Stream ecosystem metabolism is the amount of gross primary productivity and ecosystem respiration occurring in a stream, where gross primary productivity is the total carbon energy fixed by aquatic plant and algae during photosynthesis. This results in the production of oxygen, whereas ecosystem respiration involves respiration by both heterotrophs and autotrophs and results in the consumption of oxygen. Gross primary productivity and ecosystem respiration can be estimated using daily changes in dissolved oxygen, where high oxygen concentrations are associated with daylight hours when photosynthesis is occurring and oxygen is being produced. And low oxygen concentrations are associated with nighttime hours when respiration depletes oxygen more quickly than it can be replenished. The slope of the oxygen curve is how we estimate the rate of stream metabolism. When there is a positive change in oxygen over time, this corresponds to when gross primary productivity is greater than ecosystem respiration. But when there's a negative change in oxygen over time, This corresponds to when ecosystem respiration is greater. There are many naturally occurring factors that influence the rate of stream metabolism, some being light, nutrients, flow, temperature, and stream geomorphology. By altering these factors via human development, it changes the rate of gross primary productivity and ecosystem respiration and can result in unfavorable fish habitat conditions, inadequate prey production, and hypoxia. Streams in the Lower Fraser Valley are currently altered by human development and will continue to be in the near and far future. These tributaries have reduced flows and canopy cover, lower gradient and slower moving water, and greater nutrient input from surrounding agricultural and urban land use. On top of this, there are many sensitive species in the Lower Fraser Valley, two being coho salmon and Salish sucker, whose greatest environmental threats are hypoxia, reduced flows, and habitat degradation. So the anticipated research significance of my project is to first estimate current and future state of human impacts on water quality, to assess cumulative and interactive effects of nutrients, channel structure, flow, light, and temperature on water quality, to estimate parameters to populate a stream metabolism model for unsampled streams, and then to use all of this information to help identify and conserve sensitive habitats and fish populations, such as Salish sucker and juvenile salmonids. Thank you.
Great, thank you so much, Sam. In the interest of time, we'll switch right over to both Kate and Kasey, and then we'll turn to questions.
All right, hello everybody. Thank you for taking the time to come and watch all of the presentations. So Kate and I are going to be presenting on our partnering with Lower Fraser First Nations to co-produce knowledge about watershed health. I am Nkakapmach Mimameti and Acadienne, and I have a Bachelor of Science in Molecular Biology and Biochemistry. So I am going to probably be working more on the water quality side. And Kate, if you want to introduce yourself.
Hi, everyone. I'm Kate Mussett. I was born and raised in Halifax, Nova Scotia. I hold a Bachelor of Arts degree with honors in anthropology from UBC, but my work and field experience has centered more on aquatic ecology. We've partnered with MyTax and the First Nations Fisheries Legacy Fund to carry out a project focusing on biocultural indicators of fish habitat and water health in the Lower Fraser region. The project is largely inspired by the Maori Cultural Health Index, which is established in order to provide an avenue for Maori people to take on a more active role in aquatic resource management, as well as to incorporate Maori values and perspectives on aquatic health into management decisions. Representatives from the 6 partner First Nations that make up the First Nations Fisheries Legacy Fund Board, Tsawwassen, Tsleil-Waututh, Coquitlam, Katzie, Kwantlen, and Musqueam, have established a need for a similar framework in the Lower Fraser region, where place-based cultural values, perspectives, and indicators of well-being can be used to create space for multiple ways of knowing in aquatic ecosystem assessment, while also providing a more in-depth understanding of culturally and ecologically important watersheds. The Coquitlam watershed shown here is currently under discussion as a strong potential pilot location for framework implementation.
And since this isn't exactly the usual way that a lot of academic research has been done in partnership with Indigenous nations, or even just outside of that realm, our research is going to be centered around some of these key points here, which are not usually the case, I think, in a lot of co-produced research. But our most important factor that we're looking into and really considering in every step of the process is reciprocity, and understanding that there is a give and take, which also ties into humility in what we're dealing with, that we have to understand that we don't know everything as researchers, that we might think is needed in certain contexts within the watersheds. And even me as an Indigenous person, these aren't my nations, so I have to understand my positionality within this work also. And so we're probably going to be doing a lot of interviews and workshops with this work. It's still in discussion with the nations and the First Nations Fisheries Legacy Fund board members to determine whether or not this is going to be the best way to do things, especially during COVID. So we still have a few specifics to work out there. But within these interviews and workshops, we really hope to get community input on what the community themselves see as important biocultural indicators of watershed health, rather than asking them specific pointed questions, perhaps, or giving examples of other ecosystems, so we could really see what the communities and the specific communities in these watersheds see as important measures of watershed health. And we're also going to be trying to tie together or bridge, or people have said all sorts of words but to describe this process, but we're going to be really considering the Indigenous knowledge systems and Western science on an equal footing, and not having one be more of an impact in our research than the other, and understanding that both are equally valid and can work together and contribute to the research in ways that either alone wouldn't be able to do. So our goal is to hopefully tie all of this together during COVID, and to work in partnership with the nations to develop a meaningful relationship and research output. So thank you for listening to our brief presentation. If you wanted to get into contact with any of us, our emails are here, and you can also contact Andrea Reed, our PI, but if you have any specific questions about our presentation, feel free to contact Kate or I, and we also have the website link here for the Center for Indigenous Fisheries. Website is pending, but feel free to check out this information. So thank you.
Thank you all.
Thank you so much. That was a really nice intro to the work that you're planning to undertake. And so it sets us perfectly for the 5 minutes question time we have for both presentations. So please, everybody should be able to raise their hand or type a question in the chat. So you can go either way. I'm happy to moderate. Andrea, I see your hand up. Go ahead, please.
Thanks, Colette. Great presentations all around. I had a question for Samantha. For the different areas that you're going to be sampling in, does that include kind of impeded watersheds as well as unimpeded ones? Are you going to be working in spaces that have tidal flats or dams, and how do you expect that might impact some of your findings in water quality?
Yeah, so sites are going to be ranging from Vancouver mostly to Abbotsford area, and majority of the sites are going to be in agricultural and urban land use areas. And next steps are going to be going out and looking at how to meet certain criteria for higher water temperatures and lower flow velocities. So we're going to try and find a wide range of these criteria. So we want to kind of get an idea of what a low flow site might look like versus like a high flow site. So we want to, like you said, kind of look all around at impeded sites and ones with higher flows to really kind of get significant results.
Thank you.
Anyone else for a question? Otherwise, I'll jump in and ask one. Okay, I'm going to quickly then ask Kasey and Kate a question. So I was just wondering, given the situation as you highlighted of COVID making things a little bit tricky, I was just curious as to how you're planning to have the workshops. Are you going to have the communities all together, or are you as a first step trying to engage with individual communities first and then to bring them all together?
Well, pretty recently, we had a meeting with our First Nations Fisheries Legacy Fund, kind of a mouthful, but the board members, and two of the partnering nations had mentioned that within their communities, they have sort of committee meetings around the resource management and water management that they do within the community, and that these are generally pretty frequent meetings, and that people do come in and show up and participate quite often. So we're working towards possibly starting to at least develop a relationship as much as we can at the moment, because we can't meet a lot of the community members. So this is probably a way that we'll start to reach out to people and at least have the project known and introduced, and even introduce ourselves, so that we don't follow the usual tract of just researchers who come in, extract, and leave, but to prove that we are here to work together and create relationships and good research.
Yeah, and it's
Fantastic.
really nice having representatives from each of the nations, because that will largely inform who's taking part in the workshops. And it's nice to have a mediator between the academic institution and the communities that we're wanting to work with, and that are hopefully wanting to work with us.
Great, thank you so much for that. That was great. So that was our first round, right on time pretty much. So I'll pass it back to Rashid so that he can introduce our next speakers.
Now we move on to the three PhD students. So our next speaker will be Aaron Greenberg, and he's a PhD student with Dr. Murdoch McAllister, and he's going to talk to us about his work on Dungeness crab. So that should be interesting. Yeah. The next one will then be Meaghan Efford, and I looked at her bio and she described herself as a zoo archaeologist. She's a zoo archaeologist, which is an interesting one. And she's working with Dr. Willy Christensen, and her research is going to combine archaeology, ecosystem modeling, and Tsleil-Waututh science, indigenous science, to reconstruct pre-colonial fisheries in the Burrard Inlet. So it's exciting to hear that. Finally, we'll hear from Rowena Grieber, who is supervised by Dr. Marie Ugamete, and she's developing methods quantitatively to include indigenous knowledge into animal movement and habitat models. It's really nice to see so many of the research really focusing on indigenous fisheries and knowledge and frameworks. Beautiful. So thank you all, and over to you, Aaron.
My name is Aaron. I've been studying Dungeness crab up in the Hecate Strait. The Hecate Strait is in between Haida and the northern mainland. These are the different crab management areas. I'm particularly interested in up north. Dungeness crab are caught in crab pots, and these are some of them that have just been pulled up. After 6 months living out as pelagic larvae, they settle out as megalopa, and then they live on the bottom for the rest of their lives. The fact that they have a 6-month-long larval period makes looking at stock recruitment relationships a bit complicated. So we really cannot look at those sorts of questions. When they're young, they molt multiple times a year, but as they approach legal size, they start molting just once a year. And that becomes really interesting and important in a lot of my work. They are only able to be harvested when they reach when they reach 165 millimeters in size. Most of the fishery is active during the summer. So these are all of the vessels weighted by how active they were, how many traps they were pulling each week. And so they're almost all active during the summer. Then in the winter, the fall and the winter, there's a lot less fishing activity. Crabs are managed by size, sex, and season. So crabs must be male, larger than 165 millimeters in width, and hard shell to be marketed. They're measured notch or point to point on their carapace. A lot of my research has centered around two questions. Given that the fishery does not have any restrictions on catches, does the fishery harvest all of the legal sized males over the course of a summer? And how to rank some rules to avoid setting a whole bunch of crab traps on the soft shell crabs that you cannot sell, because when you pull soft shell crabs out from the water, a lot of them die. So if you just were to wait a couple of weeks for their shells to harden, then you could harvest them. So over the course of the season, this is 2004, a year of pretty high abundance. And this is 2006, a relatively low abundance year. Over the season, the fishery opens, and just over the summer, most of the catch is harvested. And then by October, the fishery basically shuts itself down. This is shown in pretty much every year. And you can see also that the effort drops really quickly towards October in pretty much every year. For the weekly exploitation rates also drop pretty quickly towards October in every year. There's different vulnerability hypotheses that I was looking at, but they're all basically turning pretty small by the fall and the winter. So to answer the first question, a lot of crabs are left and seem to be left in the water after the fishery is conducted. I mean, in 2004, we started off with like 10,000 and it drops down to about 2,000. But it doesn't go much below. They always seem to level off. This is probably a reflection on that there is some density of crabs that just cannot be harvested economically. And as the winter progresses, it gets really rough seas. So nobody really wants to go out and pursue the last crabs. We looked at 3 different harvest control rules. One that is based around fixed dates. So they would open like August 1st or July 1st. One that was based on a soft shell survey so that they would be measuring what shell conditions things are. And this is the current management regime. And one where they just didn't close. And in all years, the catch per trap haul seems to be able to be highest or near highest in the survey rule. The total catch is almost always highest in the survey rule, and the handling losses are lowest. You can also see that in some trade-off plots where you look at total catch versus catch per trap haul. The survey rule doesn't give you quite as high catch per trap haul as some of the fixed date rules, but it gives you the highest total catches and lowest handling losses. So to conclude, many fisheries are constrained by a lot more than regulations. Weather, economics, alternative fishing opportunities, and other things do constrain fishing activity. Using the soft shell survey allows the fishery to avoid handling mortality while having pretty high catches on male crabs and high catch per trap hauls. Collaboration with the Area A Crabbers Association and DFO has been really important and somewhat time consuming. If you are going to really be working with people, you should make sure you budget adequate time for all of the social interactions that are required to do that well. And yeah, the using an agent-based computer simulation model worked, but that I just put there because my advisor told me to. So to conclude, there are too many people to thank. Dan Edwards and the Area A Crab Association were really important to this project. DFO has been really important. The Canadian Fisheries Research Network has been really important. Rashid has been really important. and a whole bunch of students.
Thank you, Aaron. The next presentation is from Meaghan.
Okay, so I'm going to talk about my current focus of my PhD research, which is combining archaeology, ecosystem modeling, and Tsleil-Waututh science to reconstruct the pre-contact ecosystem state of Tsleil-Waut, or Burrard Inlet. So Tsleil-Waut is the original traditional name of Burrard Inlet. It is the singular form of Tsleil-Waututh, which is the Hunkaminam translation for people of the inlet. So the Tsleil-Waututh Nation is built around and very much centered within Tsleil-Waut, and it's a very important part of their traditional territory and their history. The two sites that I'm focusing on are Tumtumewatun, which is one of the biggest and most important sites in the inlet. It's located in what is now also known as Belcara, and Semamet, which is currently located in what is now known as Port Moody. So the main goal of this research is in support of the Tsleil-Waututh Nation's Cumulative Effects Monitoring Initiative. So all of the questions and procedures and everything like that is based in the questions and needs of that initiative and that project. So I'm establishing a pre-contact baseline food web using EcoPath, and I'm going to be supporting work to track the arc of change to the current environmental state of Tsleil-Waut using that baseline. And we're going to be investigating how the future productivity of the inlet and the contribution of seafood to the diet of Tsleil-Waututh Nation members could be increased in the future. But this is a project funded and created by a MyTax Accelerate internship with obviously Tsleil-Waututh Nation and the IOF as important partners, but Carewood Lytle as our consulting engineering partners as well, providing a lot of support and data as well. So my approach for this research is really multiple ways of knowing, working together towards a common goal. So Tsleil-Waututh science, the observations, the records, the ecosystem monitoring that has been happening over generations in Tsleil-Waututh Nation, as well as historical and archival records, mostly ships records and journal entries and things like that from early colonial contact. Zooarchaeology, which is the archaeology of animal remains specifically, and ecosystem science. So what everyone here is familiar with, but everything is working together and informing each other in a very responsive process in order to create the ecosystem model. So just for the next few slides, there are some pictures of bones, and my current research partnership agreement does not include sharing those pictures on social media. So please do not share those pictures on there. So for zooarchaeology, we take material out of the ground in what is usually a shell midden, which is a massive, usually the size of a university building, accumulation of bones, shell, and other material that is accumulated over thousands of years. And the material that I'm working with was collected in the 70s and 80s, and it was a selective collection, which means the archaeologists decide what was important to take out of the ground and take back to the lab, and everything else was left at the site. We use sieves and screens in the field to wash with water, usually just water from whatever body is close by, to remove the dirt, and then to pick out artifacts, stones, bones that the archaeologist thinks are important, beads, anything like that, and then package it all. And in the lab, we use nested geological sieves, like the photo on the far right. Usually 6.35 millimeters and 2 millimeter screens are the most common. And this is really important to note because some bones and shells and fragments of those things fall through those screens. And so we don't ever get them back in the lab, and they're not included in the analysis unless you're specifically intentionally taking that material as well. The material that I'm working with from those two sites was bagged by those archaeologists in the 70s and 80s in paper lunch bags, which are not exactly very secure and don't hold sharp fragments of bones very well. So part of this process is also re-inventorying and repackaging it, which is why this data collection phase is taking so long. but it also means that I am re-inventorying everything and getting a really good idea of what was in the ground, with the exception of what wouldn't have survived the sieving and the screening. So there are lots of different animals, but I'm pulling out a few specific ones to talk about today. There's eagle, which is very exciting as a really important bird culturally to Tsleil-Waututh and to other Coast Salish nations, but also as an important predator of herring. There's also lots of waterfowl, especially ducks, and then a whole bunch of bird bone that I cannot identify. I'm not a bird bone specialist. So if we wanted to focus on that, it would take either another archaeologist or either collagen or DNA analysis to identify those. For the mammals, there's an awful lot of deer. That's the primary bone that we're finding, is tons and tons of deer bone. There are also some either dogs or wolves, and I can't distinguish between the two personally, so we're going to try and find out which ones they are, or if it's both. There's also elk and some possible moose, both of which are very exciting because there aren't a lot of records of either elk or moose in the Tsleil-Waututh territory area, especially down by the water. There's sea otter, a lot of small terrestrial mammals, and some marine mammals, including what is possibly porpoise. It could also be dolphin, but again, we need some more specific identification. And then a lot of fragmentary mammal bone that is impossible to identify just by looking at it, which is how I've been identifying these things so far. There's also an awful lot of fish. The focus of their collection was on mammal and fish bones, but so far I've identified flatfish, spiny dogfish, herring, surf perch, and a very large amount of salmon vertebrae. We can't speciate salmon by looking at the vertebrae. You have to use either collagen or DNA analysis or something like that to speciate it. There are some people who are working on making it possible to identify salmon vertebrae just by looking at it, but it's not currently possible. And there are also salmon teeth, both the big hook teeth and the plates from the jaw bones that have the smaller, more needle-like teeth as well. But salmon make up the overwhelming majority of the fish vertebrae part of the collection. So translating archaeology to ecosystem modeling is very difficult. Not a lot of people are doing this. It's hard to establish a specific technique. So what I'm doing is taking the quantification of animal remains, the fish, the mammals, and the birds, and translating that into functional groups in an ecopath model. And I do have a draft ecopath model right now that I'm working with 81 different groups, both terrestrial and marine. It includes plants, animals, human communities, spiritual entities is coming as well at some point, but that will require more work directly with Tsleil-Waututh Nation. And Tsleil-Waututh Nation is both part of the ecosystem and as a fishery, because that is the most representative part of this process, making sure that Tsleil-Waututh Nation is represented as part of the ecosystem, part of the environment, a community in its own right, but also a fishery industry force in the ecosystem. But the archaeology gives us a good idea of what people were taking out of the system relative to what survives hundreds and thousands of years in the ground. And it talks about human use and relationships. It gives us information about changing environments. So all of that information can inform the ecosystem modeling. For example, eagle, like I said, is an important predator of herring, and herring is a really important species for a lot of First Nation groups. So knowing that relationship between all of these different groups in the ecosystem and the environment informs how it can be represented in the ecosystem model, but that's still in a draft form at the moment. So if you want to learn more about habitat change in Burrard Inlet or Tsleil-Waut, you can either follow this QR code or you can go to changeinburrardinlet.ca. As part of the Ocean Leaders program, some of my fellow students, Fanny, Roshni, and Mateus, and I created a story map of this whole process of habitat change in Burrard Inlet. And I'm really proud of it. I think we're all really proud of it, and it's really great. So please definitely check that out when you have a second.
Thanks, everybody. for attending today's talks. They've been fantastic so far. I'm going to share with you some of my initial results from my PhD on the Indigenous knowledge of bearded, ringed, and spotted seal behavior and habitat use near Utqiagvik, Alaska. I'm going to start by thanking my co-authors on this work, many of whom are the hunters who have participated in the interviews and sharing their knowledge with us. So as many as you know, there are applications of Indigenous knowledge within things like critical habitat designation and species listings that are legislated in Canada and in the US, although it is done in quite different ways actually, whereas in the US, they often focus on consultation rather than inclusion, which is inclusion is verbiage that's typically used when we think about the Species at Risk Act in Canada. And as a result of this, the consultation, I think, verbiage and focus in the United States, there's been quite a bit of conflict around species listings, particularly when subsistence communities feel that their Indigenous knowledge has not been included properly and they've not been consulted properly in this process. A great example of this is the listing of ring seals in Alaskan waters. So they were originally listed under the US ESA in 2012. A critical habitat was proposed, although never finalized, in 2014. The listing was then legally overturned in 2016 due to petitions and legal activities from groups representing subsistence communities in Alaska. This was then reinstated though again in 2018. And now we have new proposed critical habitat, which is shown here in the map, which essentially goes from the Alaska coastline all the way to the US EEZ. So it's a huge swath of the ocean. There was a petition again to have it delisted by, again, the same groups representing subsistence communities, which was overturned. But now again, there's going to be a review process of the critical habitat and the listing of the species. Now, Indigenous knowledge in Alaska has been documented in the past for quite a few different communities, but usually it's done at very large scales, looking at things like migratory movements and some hunting activity, and a little bit more recently on climate change. And this is where our project comes in. So I'm working in collaboration with the North Slope Borough, which is the governing body for 10 Inupiaq and subsistence communities in northern Alaska. And largely because there was a desire to document more specific Indigenous knowledge and do this in a slightly different way than in the past, but then also include that knowledge into habitat models to better inform things like critical habitat. So again, my work is focused around Utqiagvik, Alaska, shown here. And we're using a case study right now focused on 3 different seals. So bearded seals, or ugruk in Inupiaq, ring seals, which are Nutchik, and spotted seal, which are Kasigiak. All 3 species are important subsistence species for many communities in Alaska. Bearded seals and ring seals are a migratory species, although individuals do spend time year-round around Utqiagvik. But the individuals that do migrate will leave in the fall, in the winter, when the ice starts to form in the region, and migrate south down into the Bering Sea, and then return in the spring to the Beaufort Sea around the area of Utqiagvik. Spotted seals are definitely a migratory species. No individuals overwinter. Many of the hunters say 2 weeks before the ice shows up is when the spotted seals leave the region, and typically they do not return until all the ice is gone. Now, we've used a mixed methods approach where we do semi-directed interviews to document the Indigenous knowledge, but then use a quantitative approach to summarize and present that information that's been shared. So one of the great things about doing semi-directed interviews is that they look a lot like a conversation. So we don't have specific questionnaires, and we don't ask every hunter the exact same question, but we do ask about specific topics. And the great thing about this as well is that if hunters want to discuss other topics, they're free to move between things that they think are significant for these species. So an example of this is where we were really focused on things like ice type and ice concentrations shown here in the photos, and whether or not seals were associated with different ice concentrations or ice types, and also things like water bodies. Are they observing these seals in lagoons or in different river mouths, things like that? But what we learned through the interviews is that currents and winds were really quite important as well to the distribution and activity and behavior of these species. And these were topics that were way lower on our list of things to focus on. But so obviously through the interview process, we've discovered that these factors are much more important than we initially had thought. So I'm going to go through just a short snippet of some of the results from this. So the first 3 slides here are showing some of the distributions for these species, and darker colors represent areas where more hunters have indicated that this is important habitat for these species, or that they're frequently observed there. For bearded seals, you can see their distribution in this region is really quite wide, but there are some focused areas around Elson Lagoon and kind of between Utskavik and Monument along the coast here. When we look at ring seals, we see a little bit of a different story. This is an interesting species. The hunters basically have said that they are everywhere all the time, but we do see concentrations of them, again, in Elson Lagoon and much more along the coast here between Nuvik, which is the point, and Monument as well, and then some broader distributions down here through to Peard Bay. And spotted seals definitely have a much more spotty distribution in this area. There are large hollowouts that we see on sand spits around Tiny Island here in Admiralty Bay and Orluk Island, and that's actually an area where we consistently go for capturing and tagging of this species. We see far less use of things like the lagoon, and then also spottier use down along the coastline here. So as I've mentioned, another thing we've done is done like a quantitative summary of the Indigenous knowledge shared. And part of our process is that we do initial interviews with the hunters. And then we return, once I've interpreted the information shared from the initial interviews, and show them these types of graphs. And then it gives us time for the hunters to correct anything I may have missed, incorrect interpretation on my behalf, and also gives them an opportunity to share additional information that they would think we may not have captured during the first interviews. So in this graph, we've got along the x-axis, we have season. This is an example for ice concentration, so going from 10 to 20% ice concentrations up to 100%. And the size of the circle indicates the percentage of hunters that have shared this information. And so what we see here for bearded seals is that obviously we have a transition from the use of higher ice concentrations in the spring through the summer, which makes sense when the ice leaves. But they're more typically associated with ice concentrations of 70 to 80% in the spring and then lower through to the summer. But when we look at ring seals, they have a higher association in spring with those 100% ice concentration regions, but they also more frequently use things like leads, so which is why we see kind of this increased use in the lower ice concentration than bearded seals. And also in the winter, we had more Indigenous knowledge shared about their associations with ice in winter, because they're more frequently observed in this region than bearded seals. And spotted seals, we see, of course, a very low association because we know that they tend to migrate out of this area when the ice starts to form and return when the ice leaves. But there's infrequent observations, but some do exist of associations with ice concentrations. This is an example for shorelines and water bodies. And again, similar to before, where the size of the dot indicates percentage of hunters that have shared this information. It's interesting to me that we see with the bearded seals, and when we think back about the critical habitat designation for ring seals, that the hunters have indicated that there's increased use of nearshore waters for ring seals rather than those offshore areas, whereas bearded seals will tend to be spread throughout those two regions. Rivers and creeks is a really interesting one. What we see is that bearded seals and spotted seals are observed in rivers and creeks in this area, but definitely not ring seals. And we've done some initial interviews in 2 other communities that are along the migratory path of these species. And in Kotzebue Sound, which is just north of the Bering Strait, they actually observe ring seal use of rivers and creeks on a regular basis. So it just goes to show the difference in habitat use along the migratory route of these species. And again, as well, we see differences in kind of those inshore water bodies. So things like uses of bays and inlets or sandbars and lagoons between the different species, where spotted seals are highly associated with that type of habitat compared to bearded seals and ring seals. And this has important considerations when we're thinking about any sorts of development in these areas as well. Now, a large part of why we're looking to do this, as I had mentioned before, is the desire for the communities to have more input into things like designation of critical habitats. And so previous work that I've done on bearded seals, where we use satellite telemetry data to look and model foraging and traveling areas, has indicated that we do see foraging activity up here in Utqiagvik area. And in other regions along the coast. Now, bearded seals are notoriously hard to capture. They're huge and you only ever see one of them at a time. And so this is data for only 2 seals, which we were actually really proud of, but we now have 5 seals, which I know isn't great, but in our world, it's fantastic. But obviously, when you start to narrow in, we can see that our limited information on the species really doesn't reflect the importance of habitat or areas that the Inupiaq are finding and observing this in these species. And so clearly including Indigenous knowledge into these types of models will give us much better information about important and critical habitat for these species. So just to wrap it up, we saw differences and associations with ice formations, which can give us insights into climate change in this area, where we're seeing drastic changes in ice concentration, but also when ice is forming, and the importance of inshore habitat for things like spotted seals and bearded seals. And obviously we have implications for definitions of critical habitat for all 3 of these species. And with that, I'd just like to wrap up a thank you again to all of the hunters that have participated in this work so far, 2 of which who chose to remain anonymous, and my funders for this project as well. So thank you.
Does anybody got a question?
Everyone's presentations are great. My question is for Meaghan on the spiritual entities that you want to incorporate into your model. Maybe you can't speak on it yet because it's still in development, but I'm just curious to hear a little bit more. Like, is it about the spiritual entities of the water itself, or things like salmon, or things like thunderbirds that aren't necessarily physical animals, but are supernatural beings?
Absolutely. So right now we're thinking of all of the above, whatever Tsleil-Waututh thinks is important to represent in the ecosystem, especially related to the specific research questions. So relating to salmon, clam gardens as well, the evolution of the area. So both important terrestrial sites and entities within and related to the water. But the specific entities haven't been determined yet, and that conversation is going to be happening within Tsleil-Waututh Nation and then relayed back to me. And then we'll be working together to see how to appropriately and respectfully include them. And part of this might include a more private model versus one that is shared more publicly, depending on the specifics of the locations and the sensitivity of the information.
I have a question for you, Aaron. How do you think the change in climate and therefore climate change will affect the recruitment of the crab species that you're studying?
That is very difficult to assess. There's concerns certainly about ocean acidification, as with a lot of creatures that have exoskeletons. The difficulties of assessing it are that throughout its range, there are pretty well documented cycles of orders of magnitude of abundance. Like in California, there were decadal cycles where it was a couple of orders of magnitude larger, and then years of low abundance, and then years of high abundance. And so I'm not really sure how you would be able to separate those sorts of large dynamics from climate change on small timescales.
And my question for Meaghan concerns how to interpret what remains you find in the midden. I understand that not all of the animals that are harvested, some of them would be maybe fully utilized and very little would end up in the midden. Others like partially utilized and quite a bit of hard parts ending up in a midden. So like, how do you interpret that? And are you planning to like speak with Indigenous peoples to try to get some understanding of what proportion of like a certain animal would end up in a midden? Because that, if you're going to use all that in ecosystem modeling, it's important to understand how the midden represents what was actually harvested. And there's an important sort of number of things need to be considered in terms of that interpretation.
Yes, absolutely. And I can send you some more specific information because it is a bit of a long explanation to get really into the details. But really the straightforward answer is that if there's a lot of one kind of animal in the midden, such as salmon, then that is going to translate to specific balances in the ecosystem relating to their predators and their prey. And at every step, and on a very regular basis, I'm consulting with our representatives and collaborators at Tsleil-Waututh Nation to make sure that what I'm interpreting is representative of the knowledge that they hold. So it's clarifying and supporting each other rather than just trying to create separate information. But I'll hopefully have something more specific to communicate. And we're working together on the director's search, so I can definitely keep in contact with you regarding that information.
Rena, I really love the way that you're combining the different data sets. And so I was just wondering whether you have a way to standardize across the different hunters that you work with, that they're sort of, or are you just, is that the basis of your approach, that you assume that they evaluate the different environmental factors similarly?
Yeah, that is an assumption. In terms of the hunters that were interviewed, they're all experts and in hunting these species of seals, and so and they do vary widely in terms of where they hunt as well. And one example of this actually is that there's only 2 of the hunters we interviewed that actually hunt ring seals in the winter, so we do weight their information differently. So we say that we have 100%, for example, if both of them share information about a specific topic, whereas the other hunters might not share information in the winter.
Thank you.
Thank you. Thank you, everyone, and all the presenters for the session today. We will try to keep that format for each year, maybe each semester.